Hello, Bio 201. This is going to be a short lecture that covers the end of Chapter 13, the slides that we did not get to on our very last day of lecture because we chose to go on to 14 and cover 14 in its entirety. If we think back to 13, you'll remember that we were talking about methods of gene regulation. And so we had talked about chromatin structure, we had talked about transcriptional control, we had talked about post-transcriptional control, mainly alternative splicing, and we talked about the gene DSCAM in Drosophila fruit flies. In other words, Drosophila is the genus name for fruit flies. And we said that DSCAM is a gene in fruit flies that is one gene, but makes as many as maybe 38,000 messenger RNAs from that single gene as a result of alternative splicing. What we had not gotten to are small RNAs. What we see is that some of the genome that doesn't code for proteins codes for segments of RNA that influence the expression of the portion of our genome that does code for proteins, the 21,000, in other words, genes that make up a human being. And so it's an interesting idea and one that is a more recent discovery than many of these other mechanisms of gene regulation that we've been talking about. And so this RNA works in several different ways. One thing that it can do is simply alter the compaction of DNA, but there are two other specific mechanisms that these small RNAs can use to influence whether or not a gene is expressed. So let's look at the diagram from our text. So we see the DNA, and then we see that the DNA is transcribed into RNA, but the RNA folds around on itself, and so it's double-stranded RNA, which is unusual. So you're used to thinking about RNA as being single-stranded. Then that double-stranded RNA is chopped up into these things we refer to as small RNAs. These small RNAs can do one of two things. One possibility is they become microRNAs, miRNAs. And so microRNAs bind in a complementary fashion to messenger RNA that has been created from genes, in other words, that code for proteins, and it silences that messenger RNA. So in other words, the gene is read and you produce that messenger RNA that would go and become a protein, but the microRNAs bind to it and silence that particular gene. So it's an interesting idea, and it's one that's widely used in biotechnology today. If you want to, in an experimental animal, turn off a particular gene, then what you'll do is order these silencing RNAs that bind to that particular gene. It's also worth noting that this is the cutting edge of pest management in agriculture. And so you're familiar with the genetically modified plants that we have today. They're mainly, as we talked about, herbicide tolerant or insect tolerant. In other words, the BT plants that we discussed in Chapter 14. Well, the next generation of genetically modified crops is going to possess microRNAs that are specific to the particular pest that prey upon them. And this is great news. So as you would imagine, this would be an incredibly specific form of pest control. If you could have the DNA that codes for these microRNAs that are specific to something like the western corn rootworm, which is a big predator on corn, if that's in the corn itself, and then when the insect eats the corn, the insect dies. So in the natural state, though, just in the cells of your body, for instance, these microRNAs play a role in modifying expression of genes. Now, the other possibility for small RNAs is similar, and what we see is that a silencing RNA binds to what's referred to as a ribosome-induced silencing complex, this blue thing that we see here, which is a collection of proteins, but it's the silencing RNA that adds the specificity to this complex. And so similar to the microRNAs, this complex binds in a complementary fashion to particular segments of RNA that have been read from proteins. And when they bind, that RNA is degraded, and of course it will not be actually made into a protein. So interestingly, what we notice is that the cell has multiple mechanisms to regulate gene expression, and some of those involve RNA that interferes with the RNA that is part of the traditional central dogma biology with which you are familiar. We now go to translational control. And what we see is this, that translational control determines how much RNA is turned into a protein product. And so there are different ways of doing this. So the presence of that 5' cap that we talked about a couple chapters ago is certainly a mechanism that can influence whether or not a piece of messenger RNA is going to be read and how long it's going to be read. But more importantly, the length of that poly-A tail on the 3' end of the messenger RNA is going to determine, as we mentioned previously, how long that messenger RNA lasts in the cell, and therefore how many times it's going to be read, and therefore how much protein product is produced from the messenger RNA. You do have to remember that messenger RNA is fragile, what we refer to as labile. It breaks down fairly quickly. And this means that its lifespan is limited. It's just a question of how long that limited lifespan is going to be. A longer limited lifespan means that you have more protein. A shorter lifespan means you have less protein produced from that particular piece of messenger RNA. So the cell can easily modify the number of adenines in that poly-A tail and therefore influence how much protein is produced from that particular messenger RNA product. Next, we get to post-translational control. And so what we see is that in post-translational control, we are controlling the activity of a protein product. And so this is fairly common in the cell and in the body. And some good examples would be proteins that are activated. So there are proteins such as digestive enzymes that are found in the stomach that are very potent digesters of proteins. And so they chop up amino acids and proteins very effectively. And so in this case, it would be dangerous for the cell to synthesize them in an active form because they would chop up the proteins that are found within the cell that is producing this particular protein. Therefore, they're produced in an inactive form and not activated until they make their way out into the lumen of the stomach. Once in the lumen of the stomach, then typically a segment is cleaved from the protein chain, and that allows the protein to assume its final conformation and begin its activity. Another possibility, or actually another good example, would be insulin. And so insulin, interestingly, is produced in an inactive form, and then just like the last example that we gave, a segment of amino acids is cleaved from insulin, and that activates insulin. Insulin, as you know, is a protein hormone, also referred to as a peptide hormone, a fairly short chain of amino acids. And we see this in this simple drawing on our slide, insulin. Now you see the term proinsulin. Pro is a modifier that's included in front of a protein to indicate that it's the protein in its inactive form. So proinsulin is the insulin product before it is activated. And then what happens is we chop off this gray section of amino acids, and this that I'm circling here, of course, is the active insulin, no longer proinsulin. And interestingly, in this case, this becomes C-peptide, and C-peptide is another amino acid chain that serves as a hormone in the body. Another possibility, the last one that we'll mention here in post-translational control, is we can modify the degradation rate of proteins. And so if we speed up the rate at which they are broken down within the cell, we can, of course, control how much of that particular protein product is present in the cell. We go back at the end of our chapter here and talk briefly about mutations once again. Mutations are a very important topic in biology, and I know we've talked about them a little bit so far, so we want to revisit that topic briefly. So a gene mutation is, of course, a permanent change to the sequence of that we find in DNA. And the effects of the effects of a gene mutation on a protein can vary widely. I think that was hopefully clearly illustrated in that activity we did in class. So you could have a silent mutation which has no effect on protein activity, or, and this is more likely, you could have complete inactivation of that particular protein. So most mutations are going to be somatic mutations. Somatic mutations refer to any mutation that does not occur in the reproductive cells. Now, rarely germline mutations occur, and so these take place in the sex cells, and these would be passed on from parents to offspring. So mutations do occur spontaneously, and as we've talked about in class before, the primary means by which mutations occur is a result of an error that's made by a DNA polymerase. Now, overall, what we see is this, that the DNA polymerase is pretty good at proofreading and generally corrects errors, but the mutation rate that we observe in science overall is about 1 in a billion nucleotide pairs replicated. And so this doesn't sound like much, but you have to keep in mind that there are 3.2 billion bases present in just the human genome, which is by no means the largest of the genomes. And you're producing, our example of 2 million red blood cells per second, so that's a lot of replication. So there's a good, there's a good deal of potential for these errors to occur. Now, we also talked, if I'm not mistaken, about transposons. So transposons, when you hear transposon, think jumping gene. Transposons were discovered by Barbara McClintock, a very famous female scientist who was kind of a contemporary of Watson and Crick, maybe a little bit younger than them. And she noticed, she studied corn, and she was able to document the movement of these segments of DNA from one part of the chromosome to another, and these are widely observed in all genomes, all vertebrate genomes at least today. In fact, our human genome is, there's a tremendous percentage of our genome that's made up of transposons. In the ecology class that I teach, we look at one particular type of transposon, and just that single type of transposon makes up 11% of the overall human genome. So transposons are several segments of DNA that cut themselves out of one place, move to a different place in the genome, and reinsert themselves. Now, mutations occur spontaneously, but there are, as you likely realize, certain things in this world that can induce mutations, can speed up the rate of mutation. So these can be caused by things such as radiation, and many forms, many wavelengths of radiation can lead to mutations, anything from UV radiation, all the way to gamma radiation, which is a very dangerous form of radiation and induces mutations very, very quickly. Organic chemicals of some types can also induce mutations, and so that's why we're very careful and try to limit our exposure to certain compounds that we use, for instance, in the chemistry lab or a biology lab. Many mutagens are also carcinogens. Doesn't mean that has to be a 1-to-1 ratio, but oftentimes that's the case. So a mutagen is, of course, something that induces a mutation. A carcinogen is something that, of course, causes cancer. So environmental mutagens include things like UV radiation, and so that includes radiation that you're exposed to as you walk outside. It's a powerful, it's a fairly powerful inducer of mutations. So I've shared with you before about my mutation, my melanocortin receptor, how I have very little melanin to protect my DNA from those mutations, and so wear sunscreen every day to protect myself from that UV radiation, which seems excessive, but it's what dermatologists recommend. And then, of course, it's also the same radiation that you're exposed to in the so-called tanning beds, which are, of course, not the greatest idea, certainly, if you're interested in avoiding mutations. Tobacco smoke is something that hopefully none of us are exposed to today, but includes a number of various mutagens that are carcinogenic within it. A good test for mutagenicity is the Ames test. And so, interestingly, the Ames test involves taking a bacteria that is a mutated strain and cannot produce histidine. It has to have histidine in order to survive. And so what you see is this, as I point with the cursor here. So we have a test tube of this bacteria. We plate it out on a plate that does not have histidine, and nothing grows. Of course, that's exactly what you would expect based on what I described. But here, we take this chemical that is a suspected chemical mutagen, we mix it with these bacteria, we put it on the plate, and the bacteria grow. And so they grow because a mutation has occurred that allows them to make histidine once again. Remember, this may seem unlikely, but there are a lot of bacteria, millions upon millions of bacteria in here, and bacteria reproduce very, very quickly. A life cycle of perhaps 15 minutes. I think your book uses 20 minutes, something in that neighborhood. So just overnight, there are many, many generations that pass and many opportunities for these mutations to occur. So the Ames test is a widely used, very quick test to screen for mutagenicity. Now, UV radiation is it's a tough one, and so it's one that we're exposed to. Anytime that we go outside, you can take a look at your skin, and you can have some idea of how protected your DNA is from UV radiation. And so the darker the shade of your skin before you're exposed to the sun, then the better protected you are. And of course, it has little to do with tanning. You realize that you could, as a person with fairly light skin, make your skin dark, but that's not what we're referring to. We're referring to the amount of melanin that you have in your skin to begin with. Here's the issue with UV radiation. It causes these things referred to as thymine dimers. So here's a thymine and here's a thymine in the double helix of DNA, and you can see this covalent bond that's formed between them. There should be no bond there, as you well know. Rather, this thymine should be bound to this adenine, this thymine to this adenine, and everything should be fine. But this thymine dimer causes a significant problem, causes a kink in the sugar phosphate backbone, and during DNA replication, it's likely to cause a misplaced base. So UV radiation is such a powerful inducer of mutations that we can use it to sterilize an area where we do research. And so at Asbury on the biology department floor there, you're probably familiar with the prep room. And if you walk in that prep room, which is right across from both of the lecture rooms that we utilize on the other side of the hall, there's a large metal structure referred to as a hood. And so you can turn on a UV light there, which would be similar to tanning bed light, leave it on overnight, and it kills all the bacteria in the hood because it induces mutations in those bacteria so quickly that they die, and so all of them are gone by the next morning. So the good news is that in eukaryotes, most of the time these dimers are removed by repair enzymes. Now, sooner or later, a mistake will be made, and so that's where skin cancer comes from, and that's why it's ideal to limit your exposure, especially depending on the shade of your skin, to UV radiation. Now, there are conditions like xeroderma pigmentosum in which those DNA repair enzymes are deficient and don't work like they should. So this is a picture of a young man who has xeroderma pigmentosum. And so you're very susceptible to skin cancer if these DNA repair enzymes are not functioning correctly, because you pick up these thymine dimers very quickly, and those lead to mutations in the absence of those proficient enzymes. Now, what we see is this, that mutations can occur in different ways. Most mutations will be point mutations. And so a point mutation is a single change in a DNA nucleotide. And so it may change the codon from one to another and specify a different amino acid, but it may not. If you think back to that activity that we did in class, as I mentioned a few minutes ago, that was a silent mutation in which there is no change in the amino acid that is included. Now, the effects, if it does affect the protein, then the effects may vary. It's quite possible that the protein will be non-functional. It's possible that the protein will have reduced functionality. Now, it's also possible, and we'll look at an example on the next slide, that it could have increased. It could have an increased functionality. And so, remember, our understanding of biology is that all alleles come from mutations. And so, the alleles that we see in humans will primarily come from these point mutations. I was talking to a friend who is a molecular biologist, and David, and he was telling me about dogs. And so you're familiar with dogs that have short legs, like dachshunds and chihuahuas and so forth. And he was pointing out a paper that he had read in which those short legs come from a single mutation that occurred in the domestication of the dog from the wolf. And so that apparently was a larger mutation, a transposon that was moved from one place to another. But in any case, mutations can lead to new alleles. If I use myself as an example, then my mutated melanocortin receptor is a single change in a DNA nucleotide. By the way, we refer to that as a single nucleotide polymorphism, an SNP. SNP. And that's why I have red hair instead of dark skin and dark hair. Now, another possibility, and this is a much more serious type of mutation, is a frameshift mutation. So in a frameshift mutation, we are inserting one or two nucleotides into the code. So take a look at this example from our text. What we see is that your book is using a series of a sentence of three-letter words to illustrate this idea. So normally, the sentence says, "The cat ate the rat." However, in this case, we are deleting the C here. And so if you delete the C, then something happens that's referred to as a shift in the reading frame. And so now, "the" makes sense, but there should be a C here. It should be cat, but it's gone. So now it reads "atta," T-E-T-H-E-R-A-T. So it doesn't make any sense. And so that's exactly what happens when you add or subtract one or two bases from the genome. It messes up the reading frame. And so frameshift mutations are very serious and likely to impact huge stretches of DNA, because instead of just changing one codon, you're changing just perhaps thousands of codons. The same thing applies after this insertion. So we are simply inserting a C right here, and you can see that it also shifts the reading frame. Now, the very last thing we're going to talk about in this chapter is the example of sickle cell anemia and a point mutation in hemoglobin. And so what we notice is that sickle cell anemia is a disease that occurs very widely in areas of the world where malaria is common. And it's caused by a mutation in the hemoglobin, the gene for hemoglobin, and it is an SNP, a single nucleotide polymorphism. And so what we notice is that our book is showing us the normal hemoglobin gene with the normal sequence of amino acids. And actually, our book is also showing us, look at this. So our book is changing this G to an A here and illustrating the idea that we illustrated with the little exercise in class, once again, where we can change the last base in the codon oftentimes and not change the amino acid that's added. But what our book is showing us here is that we're, in this case, we are changing not the last base in a codon, but the middle base in the codon. And so that does change, as you can see here, the amino acid that is incorporated. And then your book is illustrating here, if we change the first base in this codon, it actually stops the protein at that particular point. So it illustrates nicely this idea that depending on where the mutation falls, we can have differing effects on the protein. Now, we do want to point out that in the case of sickle cell anemia, there are different types of sickle cell anemia, but they're all caused by an SNP, a single nucleotide polymorphism, a change in one base. And what we see is that this is a normal red blood cell, but people that have that single nucleotide polymorphism have red blood cells that look like this. They are sickled. This is extremely problematic, and it's very difficult for these red blood cells to carry oxygen and to make their way through the smallest blood vessels of the body. In fact, very sadly, the expected lifespan of individuals that have sickle cell anemia without medical intervention is about 4 years, depending on the particular mutation that they possess. We can ask the question, why is this particular mutation so common in the world today? And the answer is that this mutation is a homozygous recessive mutation. In other words, you do not have blood cells that look like this unless you have two recessive alleles. However, if you're heterozygous for this mutation, if you have one normal allele and one recessive allele, it's not a case of complete dominance. Your red blood cells are a little bit different and you're resistant to malaria. So what we see is that for a long time in human history, that heterozygous state has been selected for because these people suffered and didn't reproduce as a result of sickle cell anemia. These people didn't reproduce as a result of malaria, but the heterozygote, which is not shown here, was selected for because they neither died from sickle cell anemia nor died from malaria. And our book is illustrating that point here. So here we have the frequency of malaria, and here we have the frequency of that sickle cell allele. And those maps correspond very nicely because of this selective process for this allele in areas where malaria, a disease, of course, spread by a mosquito, is present. At the very end of our chapter, we talk briefly about the development of cancer, carcinogenesis. And so we've talked about this fairly thoroughly before, but we'll go over it briefly here once again. And we've said that there are two types of genes that play a role in initiating cancer. A proto-oncogene, which normally stimulates cell division, but when it's mutated, it overstimulates cell division, and then a tumor suppressor gene, for instance, the p53 gene that we talked about, which is responsible for inhibiting cell division, particularly if the cell doesn't pass one of the checkpoints that occur in the cell cycle. What we see here is a mutated tumor suppressor gene. And what's happening is that we have an inhibiting growth factor that binds to a receptor. We have a signal cascade here, protein A, protein B, protein C, that activates a transcription factor that binds to the DNA, bringing about the production of the tumor suppressor protein. However, the DNA is mutated, and so the tumor suppressor gene that's produced is unable to stop the uncontrolled division of the cell and initiate apoptosis. And so, as we mentioned, over half of all human cancers are tied to that tumor suppressor gene that is referred to as p53. Lastly, we have a mutated proto-oncogene, and so therefore a mutated proto-oncogene is an oncogene. And so we have, in this case, the stimulating growth factors that bind to a receptor. We have a series of proteins in this signal cascade. We have the transcription factor. And then we have the production of an oncogene. It's supposed to stimulate the division of the cell moderately, but it doesn't. It overstimulates the division of the cell at an extremely rapid rate. So this brings us to the end of chapter 13. Please let me know if you have questions on any of this material in chapter 13 as you prepare for the final.